Hi everyone, this is Unit 6 of Neuroscience for non-majors, and unfortunately I had to get an emergency intestine surgery about 2 weeks ago, and that's why we haven't had class recently because I've been in recovery, but I've tried to unfortunately, I've had to cut some material this semester. I've had to cut the part 2 of the learning unit and the neuropharmacology unit, but today is going to just be a general overview of the different kind of sensory processing systems, as well as kind of how we move. So I'll share my screen. All right, so yeah, we're going to cover visual information processing, audition, olfaction, and taste, as well as somatosensation and pain, and movement today. So I guess the world as we see it is a result of cells in our eyes processing photons. This is a human eye, and when light goes through the eye and hits the retina, they're absorbed by, the photons are absorbed by photoreceptor cells called rod cells and cone cells. And rod cells contain a protein called rhodopsin, while cone cells contain a protein called photopsin. And these proteins are what process the photons arriving at the retinal cells and cause the retinal cells to fire an action potential in the presence of light. And we have 120 million rods and 6 million cones. Rods are located here on kind of the sides of the retina, while cones are mainly located here in this little dimpled portion called the fovea. And the fovea is the area of highest visual acuity, and it's kind of directly in the center of our gaze. And okay, so while rods are about 1,000 times more light sensitive than cones, rods do not produce color vision, but cones do. Rods are basically activated in the presence of light, and they're kind of responsible for detecting either presence or absence of light, so black and white vision. Again, the photons are absorbed by these rhodopsin proteins, and the process of the photons hitting these rhodopsin proteins causes the rod cell to fire an action potential. Cone cells, on the other hand, are the reason we have color vision, and there are 3 types of cone cells that either that can absorb short, medium, or long wavelengths of light, red, green, and blue. And the photons are processed by the photopsin proteins in the cones and allow the cone cells to fire an action potential. And the combinations of activations of the cone cells are what allow us to see all of the different, the whole variety of colors that we do see. So these retinal cells then activate cells of the optic nerve, which takes this visual information into the brain. Now, in the brain, when the eyes take in information from their surroundings, the right eye's representation of the left half of the world, along with the left eye's representation of the left half of the world, are sent to the right hemisphere of the brain to be processed, while the representations of the right side of the world in either eye go to the left hemisphere. The right side of the world in either eye go to the left hemisphere of the brain to be processed. And this information then goes to a part of the thalamus called the lateral geniculate nucleus. And each cell in the lateral geniculate nucleus are abbreviated as LGN, so each cell in the LGN is monocular, which basically means that each cell can only process information about the world that it receives from one eye or another eye, but not both eyes at once. And the thalamus itself that contains the lateral geniculate nucleus is, as we learned before in previous lectures, is a kind of sensory, is kind of a gate for sensory information, and it processes the information before sending it out again into like appropriate areas of cortex. So then after making it through the LGN, the visual sensory information is sent to the fourth layer of primary visual cortex, which is abbreviated as V1, which also has monocular cells in the fourth layer. And then from layer 4, it goes up to actually to layers 2 and 3, where the interconnectivity there allows for binocular cells now that can compare information from both eyes within an individual cell. And then layers 2 and 3 project down into also binocular cells in layers 5 and 6. And the projections from layers 5 and 6 lead out of the primary visual cortex. So binocular cells are really important for judging locations and distances of objects, as they compare the positions of objects in one eye to the positions of those same objects in the other eye. Like, as all of us know, by closing one eye and then closing the other eye, the positions of everything are shifted. So as we learned in a previous lecture, the brain is divided into the frontal lobe, parietal lobe, temporal lobe, and occipital lobe. And the occipital lobe is what contains the primary visual cortex, or V1. And so kind of the entire occipital lobe, as well as parts of the temporal lobe and parts of the parietal lobe, are all dedicated to vision, or combining vision with audition, or combining vision with like somatosensory information. So again, back from layers 5 and 6 of V1, the visual information then projects into either the dorsal stream or the ventral stream. So the ventral stream basically takes the visual information into the temporal lobe, and it's used there to identify objects and animals and people and stuff like that by combining information about shape, color, and sound. Auditory cortex is also located there. So yeah, so however, faces actually have a separate area called the fusiform face area on the bottom of the temporal lobe, where they are represented and recognized kind of separately. Then the dorsal stream takes visual information into the parietal and then into the frontal lobes, and it basically is responsible for carrying visual information about the location and motion of objects, and putting together how an object can be interacted with, as well as where it is and how it's moving. And this is why it goes to the parietal and frontal lobes, because it needs to combine visual information with somatosensory and motor information and plans, and that's where the somatosensory and motor cortices are. Okay, now we'll get into audition, or hearing. So what we hear in the world is a result of how we process sound waves around us. So first, this is my kind of a bit poor illustration of our external ear, but basically sound energy is gathered by our external ear into the eardrum, which is also called the tympanic membrane. And the external ear is actually designed to amplify sound frequencies that include, it's designed to like amplify the frequencies that include our most important speech sounds, which is interesting. So when there's this pressure then on the tympanic membrane, it causes these bones, like the stapes, and to move, and basically then all of that is causes the oval window also to then move, and all of this kind of sound energy is then focused onto the oval window. which is basically the oval window is the entry point into the inner ear, the cochlea, from the external ear. And the cochlea of the inner ear is responsible for converting the sound energy into neural impulses. And now we'll find out how the cochlea does this. So the cochlea of the is basically this coiled up structure that has 3 fluid-filled chambers inside called the scala vestibuli and the scala tympana, which both contain a fluid called perilymph, as well as the scala media, which contains a fluid called endolymph. So when that oval window is hit by a sound wave, these fluids are all disrupted. And the fluid movement also causes the emotion in this membrane called the basilar membrane. And here, the basilar membrane is kind of like folded like this, but we can look at it like this, where basically the basilar membrane also moves in a wave-like manner, and different parts of the basilar membrane are kind of more attuned to certain frequencies compared to other frequencies. So wherever the wave motion is kind of greatest, that's like the frequency that's arriving, and the basilar membrane has the greatest movement in that area that corresponds to that frequency. And when that happens, when the basilar membrane is disrupted in that way that corresponds to the frequency that that is being received, it basically disrupts these things called hair cells in that area that sit just above the basilar membrane. So on these hair cells, there are microscopic hairs called stereocilia, which are disrupted, and this causes ion channels in the hair cells to open, and ions flow through, and this generates an electrical signal that releases neurotransmitters, which then takes the auditory information into the brain to be processed. And now this is kind of a bit of an inaccurate illustration here, but if you want to know about how these specific ion channels on the hair cells actually work, they have a lot more to do with like the stereocilia than I'm showing in this illustration. It's a bit of an inaccurate illustration. Again, come talk to me and I'll explain it to you, because I think it's a very, very fascinating process, these particular ion channels. But yeah, just take this particular illustration with a grain of salt, but for just like a general understanding, it serves its purpose. Okay, so yeah, so the hair cells then release neurotransmitter. Now the information is, now the sound waves have been converted into neural impulses. So the auditory information goes to these things called olivary structures, or olives, which are named because they literally look like olives. And in a part of the brain called the medial superior olive, or abbreviated as MSO, interaural timing difference is computed. And the way that happens is basically having MSO cells that receive inputs from both the left ear and the right ear, and they're arranged so that longer axons coming from the right ear's cochlea are paired with shorter axons from the left ear's cochlea, and vice versa. And you can kind of think of these MSO cells as like coincidence detectors. So think of them as like they only activate when they're getting simultaneous inputs from the right ear axon and the left ear axon. And now, how does this compute interaural timing difference? For example, in this diagram, if a sound arrives at the left ear first, right? Then it arrives at the right ear. What's going to happen is that the longer left axon will be activated, and during that time, the sound will be able to reach the right ear, and then it will activate this shorter right axon. So the coincident activation will arrive at this neuron E in the MSO. The simultaneous inputs on the MSO neurons will arrive on this neuron E, and it will have this very strong response. And the brain will know then that when this neuron E is activated, it means that the sound was coming from the left side. So neuron E is like a left ear leading neuron. Similarly, if the simultaneous inputs arrived at neuron C, it would mean that the sound was arriving from like directly in front of you, because it took the same amount of time to reach it took the same amount of time to activate this and the same amount of time to activate this. So these two activate, get the strongest response from neuron C. And then also, if the inputs arrive at neuron A simultaneously, it means that the right ear was, the sound was coming from the right, because it took this, the time it took for it to activate the left axon, and was the amount of time it took for it to kind of like travel down this long path, and which means that it arrived here first because the impulse took like more time to arrive at neuron A, and give it the same input as the other. So I hope that makes sense. The MSO stuff is a bit complicated, but again, ask if you're confused about anything. Now the LSO, the lateral superior olive, is a lot easier. For the purposes of this class, you can just think of it as the side of the LSO that is the same side of the head as the sound source will be more excited, and the opposite side will be more inhibited. And this is how it kind of computes interaural intensity difference. So the information from the MSO and the LSO goes to the inferior colliculus in the midbrain, and then it goes to the medial geniculate complex in the thalamus, and then from the thalamus it goes to auditory cortex, which actually contains the topographical map of kind of the cochlea, and it can be integrated with other information from the brain. Now we'll move on to olfaction and taste. So we smell because of particular chemical combinations that activate particular receptor combinations in our noses. And these chemicals are a wide range of airborne stimuli called odorants, and they interact with receptor neurons that are on this inner lining of the nose, which is called the olfactory epithelium, which is it's inside your nose. And the axons from those receptors basically project across a region of the skull called the cribriform plate into your brain, and they synapse onto a part of the brain called the olfactory bulb. And neurons in the olfactory bulb then project to a cortical region in the temporal lobe. And these projections, called the olfactory tract, are actually, they're the reason why smell and episodic memory are highly linked to one another. Remember, the temporal lobe contains the hippocampal formation. And so another thing that's interesting is the olfactory system actually doesn't need the thalamus to go to a cortical region, and the projections go directly to this part of the temporal lobe called the piriform cortex. And the piriform cortex is also interesting. It only has 3 layers, so it's called an archicortex. Neocortex, by the way, refers to the cortical areas with 6 layers. So the piriform cortex that only has 3 layers. But the olfactory information does go to the thalamus eventually, even with this sort of cortical pit stop. And from the piriform cortex, it goes to the thalamus, and then it goes to targets like the hypothalamus, which controls hormone secretion, and the amygdala, which controls like fear response and emotional response, as well as other areas of neocortex. So we still actually don't know if there's a topographic mapping in piriform cortex or the olfactory bulb for this smell information. And there's a lot of ongoing research into the nervous system's representation of olfactory information in general. Another thing that's interesting is it's interesting how concentration of odorants actually elicits different responses from us. So for example, the odorant molecule called indole at low concentrations smells like floral and nice, but at high concentration it smells like rotten and putrid, and very unpleasant. All right, now taste. So when we eat food, the chemicals of that food called tastants, like odorants, tastants, kind of aggregate in these little like grooves between sticking out parts on the taste buds of our tongue, and the sticking out parts are called taste papillae. And so the tastants aggregate and they activate these things called taste cells, which are also in the taste buds. And the taste cells then are able to then kind of encode information about the chemicals they're interacting with, and they're also able to activate like salivation and things like that, or trigger gagging and regurgitation for noxious substances. But anyway, so taste cells synapse with sensory neurons in different cranial nerves, which then take the taste information to the gustatory nucleus in the brain stem. And then from there, the information goes through a part of the thalamus called the ventral posteromedial nucleus. And then finally, from the thalamus, the information goes to various areas of neocortex, and particularly interestingly, it goes to this part called the orbitofrontal cortex, and that specific connection is related to the conscious motivation to either eat or not eat a food. And orbitofrontal cortex is a really interesting area of the brain that has to do with like task switching, like switching your focus from doing one activity to another activity. And obsessive compulsive disorder is also related to kind of like issues with orbitofrontal cortex. So that connection is very interesting. Projections also go to the hypothalamus and amygdala, like with olfaction. So there are 5 classes of taste receptor molecules that kind of represent tastants in the salty, sour, sweet, bitter, and umami categories. For example, salty or sour tastes are caused by like ionic tastant molecules like salts and acids. So their corresponding kind of receptor channel proteins on certain, they have these corresponding receptor channel proteins on very specific subsets of taste cells. So the taste cells themselves are like more kind of like biased towards certain like tastes as opposed to other tastes. And there's actually a mapping on the tongue of these taste cells where sweet, umami, and salty biased taste cells are towards the tip of the tongue. And this is probably because those tastes elicit pleasure, and those tastes are also correlated with foods high in carbohydrates and amino acids. So that's why they're at like the initial point of contact, the tongue. Where sour and bitter taste sensitivity peaks on the sides and on the back of the tongue. And this is because sour and bitter tastes are often warning tastes of noxious substances, and with enough potency, they can elicit gagging and other protection reactions from the body because of this positioning on the sides and the back of the tongue. All right, now let's get into somatosensation and pain. So the somatosensory system is responsible for the sense of touch and everything associated with it, like pressure, temperature, pain, itchiness, etc. And it kind of has three main components. One that transmits information about touch, vibrations, and pressure. A second subsystem responsible for proprioception, which is our ability to sense muscle, tendon, and joint position in space, as well as a third subsystem responsible for conveying information about pain and temperature. So the skin has different amounts of sensitivity depending on its location. For example, on your fingertips, you can identify two different points of contact if they're only 2 millimeters apart. But on the upper arm, you can't really perceive two different points of contact as being distinct until they're around 40 millimeters apart. And neurons that receive information from the skin and other organs are considered afferent neurons, while neurons that send information out the other way to the organs are called efferent neurons. And rapidly adapting afferent neurons called Meissner afferents, or Meissner afferents, I'm sorry. They are basically all over the hand, and because they're rapidly adapting, it means they basically fire rapidly when a stimulus is first pressed against the skin, but then they fall silent if the stimulus stays there. And then they're also slowly adapting afferents. An example would be Merkel cells, and those are in the fingertips. And they, on the other hand, generate a sustained response with continuous stimulation. So these cutaneous nerve cells are in a category of mechanoreceptors or mechanoreceptors, and they open ion channels in the presence of pressure or distortion of the skin. So Meissner afferents are really good at conveying information about textured objects that move across the skin, while Merkel cells are able to identify kind of distinct stimuli, even if they're very close together. And then there are also other afferents called Pacinian afferents, which are really good at sensing high frequency vibrations, while another type of afferent called the Ruffini afferents they respond really well to kind of stretching of the skin. So these all have cutaneous receptors, and they provide information about external stimuli. Proprioception, basically proprioceptors are kind of a different class of mechanosensory neurons, and they're responsible for giving detailed and continuous information about limb position and position of other body parts in space. And examples of this are for example, there are these joint receptors in and around finger joints, and they activate when a finger joint reaches its limit of motion range. And there are also sensory afferents that are wrapped around the central fibers of skeletal muscle spindles, and they open ion channels when the muscle is stretched. And the muscle spindle is innervated with two different kinds of nerves, primary and secondary, and the primary nerve endings transmit information about velocity and movement direction, while secondary nerve endings provide information about static limb position. So cutaneous and proprioceptive information travels through the dorsal horn of the spinal cord and up the ascending spinal cord pathways to the brain that we learned about in a previous lecture. And so in the brain, the information travels through the pons and ventral posterior thalamus and the midbrain to the primary somatosensory cortex. And the primary somatosensory cortex contains a map like motor cortex with parts directly corresponding to feet and legs or forelimbs or face or etc. So some areas of somatosensory cortex correspond more to kind of cutaneous stimuli, while other areas respond more to proprioceptive stimuli. And then from the primary somatosensory cortex, information either goes to the motor cortex or it goes to the secondary somatosensory cortex. And from the secondary somatosensory cortex, it goes to areas like the amygdala and the hippocampus. And information also goes back to the thalamus and the brainstem to modulate further incoming somatosensory information. Now, nerve endings that initiate pain sensations are called nociceptors, and they arise from those dorsal root ganglia cell bodies. And there are two different kinds of pain nerve fibers ascending the spinal cord. There are A delta fibers, which are super myelinated in order to convey urgent, super fast, sharp shooting pain signals, while C fibers, then, which are less myelinated or unmyelinated, convey dull aches that take longer and cause a delay. Noxious heat stimuli and capsaicin. Capsaicin is the compound found in like chili peppers, the spiciness. They activate the same type of receptor, a receptor called TRPV1, which is found on both A delta and C fibers. And it belongs to a family of transient receptor potential, or TRP or TRP channels that are sensitive to heat and cold. And the TRP channels are ion channels that open when so the TRPV1 channel, for example, opens when either capsaicin binds to it or when there's a lot of heat present, and it allows sodium and calcium through, generating an action potential in those nociceptive fibers. So then the fibers travel up the dorsal horn of the spinal cord. On the opposite side is the mechanosensory fibers, and up through the brainstem, and then they transmit information to the thalamus. And so sharp shooting pain information goes to the ventral posterolateral nucleus of the thalamus before traveling to somatosensory cortex, while the second pain, the duller ache, goes to the amygdala, and then from there it activates the periaqueductal gray in the brain, which activates this suppression system. And what happens in the suppression system is that the periaqueductal gray activates this area of the brain called the raphe nuclei to release serotonin back to the dorsal horn of the spinal cord. And these serotonergic neurons stimulate and excite neurons in the spinal cord that release an endogenous opioid called enkephalin. And when enkephalin is released onto pain neurons in the spinal cord, those pain neurons are inhibited, and the enkephalin prevents them from then sending the ascending pain signal, so it suppresses the pain. Now, this whole system of like the ascending pain information and this pain suppression system is all part of this pain modulation system called the anterolateral system. All right, now let's get into movement. So we learned about lower motor neurons in a previous lecture. But today we're going to talk more in depth about like upper motor neurons and lower motor neurons and things. So upper motor neurons project descending pathways from primary motor cortex and the brainstem to the spinal cord. And in the brainstem, these upper motor neurons are conveying vestibular information and helping orient the eyes, head, and body, or controlling posture and navigational movements. So the cerebellum, I can show you the brain picture again, maybe. If you remember, the cerebellum is here. It calculates the error, the motor error, or the difference between an intended movement and an actual movement, both in real time and correcting motor behavior over time to minimize the error. And so how does it do this? It's divided into 3 parts called the cerebrocerebellum, the spinocerebellum, and the vestibulocerebellum. They're kind of self-explanatory, but the cerebrocerebellum receives all sorts of inputs from cortex through intermediary neural ensembles called the pontine nuclei. And the spinocerebellum receives a bunch of sensory information from the body, and has actually, the spinocerebellum is very interesting because it actually contains a what's called a fractured topographic map of the body, where every tiny area of the body is repeated multiple times in different spatially located clusters of cells. Yeah. Anyway, then the vestibulocerebellum receives projections from the vestibular nuclei in the pons, and we learned about the pons in an earlier lecture. So the cerebellum has a very distinct setup of neurons. Basically, inputs from the brainstem and spinal cord synapse onto neurons called granule cells, which are in the cortex area of the cerebellum. And these granule cells have axons that are shaped in a very specific way. They're shaped like a T, and they're called parallel fibers, and they synapse onto the dendrites of neurons called Purkinje cells, which are underneath the cortex layer of the cerebellum. And Purkinje cells have, like, even more than my drawing, but like these enormous dendritic trees, and they make contacts with many, many, many parallel fibers. Just like each parallel fiber also can stretch out and make contacts with many, many Purkinje cells. So the Purkinje cells, they synapse onto and send inhibitory GABA, they release GABA onto deep cerebellar nuclear cells. And those deep cerebellar nuclear cells generally go to the thalamus, which transmits information then to motor cortex. I didn't put this in my illustration, but it is important to note that there are also climbing fibers, which come from a part of the brain called the inferior olive. And those climbing fibers kind of wrap around the Purkinje cells. And they modulate they regulate and modulate the connections between the parallel fibers and the Purkinje cells. And another thing that they do is that these deep cerebellar nuclear cells receive excitatory inputs from those climbing fibers. So the deep cerebellar nuclear cells, which again go to the thalamus, are receiving inhibitory inputs from the Purkinje cells and excitatory inputs from these climbing fibers, which also regulate the interactions between the Purkinje cells and parallel fibers. So so the basal ganglia, which we learned about when we learned about this last lecture, we know it inhibits unwanted or unintended movements through the indirect pathway, while encouraging wanted and intended movements through the direct pathway. And it sends information to motor cortex through the ventral anterior and ventral lateral nuclei of the thalamus. And remember also, again, that motor cortex has a topographic map of the body, where, for example, if you stimulate the tongue part of motor cortex, your tongue will move, or your left hand finger, then that will move, and so on. Okay, yeah. So this is an illustration from from a previous lecture, but we learned about lower motor neurons. They're in the spinal cord, and they receive inputs from the brain through descending pathways made up of upper motor neurons in the brainstem and cerebral cortex that then synapse onto other lower motor neurons beneath them in the spinal cord, and then they send their axons to skeletal muscles. The motor neurons in the spinal cord send their axons to skeletal muscles through the ventral root. And but the lower motor neurons also actually receive input from other local circuit neurons. So like inside the spinal cord, there are going to be other local circuit neurons and intermediary neurons. And those intermediary neurons are going to be also receiving inputs from sensory neurons in the local circuit of the spinal cord. And so what this means is that actually, even if you disconnect the brain from the spinal cord, you can still get skeletal muscle contraction if you have this local stimulation that doesn't need the brain. And so here we have an alpha motor neuron, but there are actually two types of motor neurons found in the ventral horn. Where basically, and that synapse with muscle fiber. So the alpha motor neurons are very large, and they synapse with muscle fibers that generate like very strong like forces needed for movement. Well, there are also other smaller gamma motor neurons that synapse onto more specialized muscle fibers called muscle spindles. We talked about muscle spindles earlier, I think, that have that have the sensory receptors along with force generating fibers. Now, the last thing that I want to talk about is the neuromuscular junction. So at the neuromuscular junction, those motor neurons release a neurotransmitter called acetylcholine, and the acetylcholine binds to ionotropic acetylcholine receptors on the muscle cells, and this allows sodium ions to flow into the muscle cell, and this electrical signal actually causes the muscle cell to have an action potential. And that action potential causes proteins in the muscle cells to pull on each other, and the entire then skeletal muscle contracts. And when skeletal muscles contract, they pull on your bones and then you move. Okay, so these are the references from this week's material. And for the homework, actually next week is going to be kind of a computational neuroscience and a class where we'll talk about like artificial intelligence and brain simulation methods and brain-computer interfaces and things like that. And we'll also end then with a discussion of science fiction stories that address these topics. And I'm still reading through and trying to find the best science fiction stories that I want to assign for you guys to read, but I'll get those to you like very early in the week so that you have plenty of time to read them before the discussion. And that's it. Hope you have a nice week, and yeah, I'm sad that we have to cut. Excuse me, the neuropharmacology stuff. But yeah, all right. See you.